central dogma of molecular biology begins with genotype. The DNA contains all of your genotypes, and transcription allows RNA copies to be built, which can then be translated into proteins, which are secreted from the cell. Notice that there are more steps. The internal stimuli and the transcriptional stimuli affect the DNA, which leads to transcription. You can see the cell nucleus here. The DNA is contained within the cell nucleus and the cytoplasm without, outside. The RNA here being degraded over time. Translation, activity, modification, and degradation. Protein has activity, it can be modified, and it can degrade. And it leads to a phenotype. It's the thing that you see on the outside. Brown hair, blue eyes, white skin. The concept of a gene, a fundamental physical and fundamental unit of heredity, that a DNA sequence located on a specific site of a chromosome, which encodes a specific functional product. RNA, protein. This is from Wikipedia, so you know it's a good source. Continuing with the concept of a gene, the genome, the total genetic material of a living organism. Genes are 1.5% genome. Gene-related DNA is 36% genome. And the intergenic DNA is 62.5% genome. So what do we mean by this? Well, there's exons, which are coding portions of the DNA, and there are introns, which are non-coding portions of the DNA only found in eukaryotes. Coding equals the transmission into mRNA. So only the exons turn into mRNA. So we have these vast portions of introns which are non-coding. Why would we have these things, would you ask? How would this work? You start with DNA. You have promoter region, exon 1, intron 1, exon 2, intron 2, exon 3, intron 3, exon 4. You have transcription which occurs. So you have the primary transcript of pre-mRNA here, and you lead to alternative splicing. So you have a mature transcript of mRNA. They've taken the regions that they wanted, and then it leads to protein synthesis of amino acids. So the advantage of the eukaryotes in having these introns is that they can shuffle around the order of their exons, leading to more proteins, more variety of proteins with the same DNA strand, because the order matters. So then this leads to gene expression, the process by which a gene gets turned on in a cell to make RNA and proteins. So it's measured by looking at the RNA or the protein made from the RNA. An expression of certain genes and or an increase in their expression can be related to contaminant exposure. So note, the upregulation of RNA does not necessarily mean upregulation of protein production. Think about that. What we've got here, we've got DNA, a gene, transcription leads to RNA, translation leads to protein. Protein leads to you.